Dominant epistasis with another example. Second example, they are ends containing white color hairs or leg or hairs. These hairs are expressed by inhibitory genes. So the dominant epistasis with another example in hens with inhibitory genes. See here, it is the parental generation. Ends are containing capital I, capital C genes. These are in homozygotic condition. They containing white color hairs called white leghorn hens. And the next one, recessive ends are containing small I, small c with white plymouth horn ends. Here it is, from white leghorn, the capital I gene act as a epistasis gene, and small capital C, small I, small c. These three genes are act as a hypostasis gene, mean in presence of capital I gene, capital C, small I, small c, these three genes cannot express their own color or own character because these characters are suppressed by capital I gene. So these three genes are called hypostasis gene, and epistasis is in dominant position. The dominant position epistasis genes are capital I, capital I. If capital I is in homozygotic condition or in heterozygotic condition, they can dominate on all hypostatical genes. They can dominate on all hypostatical genes. So compulsory epistasis gene is in dominant position in the hens. From white leghorn, the gametes are capital I, capital C. From white plymouth horn, the gametes are containing small I, small g genes. These two gametes are fertilized to form F1 generation. To form F1 generation. The F1 generation ends are expressed white color leghorn ends, but these are in heterozygotic condition. See here it is, the capital I is in heterozygotic condition. Heterozygotic condition means the capital I combines with small I. It is in heterozygotic condition. If homozygotic condition means capital I is in homozygotic condition, mean the dominant epistasis genes are in homozygotic condition or in heterozygotic condition. They can dominate or they can mask on all hypostatic genes. Here it is, what are the hypostatic genes? Capital C, small I, small c. These three genes are act as a hypostasis gene. The F1 generation containing white leghorn ends. These are crossed with similar type of white leghorn ends. Same F1 generation cross with another same F1 generation to form F2 generation. The F2 generation phenotypic ratio generally 9:3:3:1 is changed into 13:3. 9:3:3:1 change into 13:3. See, we observed in F2 generation is F2 generation in Punnett checkerboard. One parental white leghorn ends produced 4 types of gametes. They are capital I, capital C. See here it is one gamete. capital I, small c, capital I, small c, small I, capital C, small I, capital C, small I, small c. Similar, these are the 4 types of gametes developed from the one parental generation of F1. These are crossed with similar parental generation of F1. They also produced 4 different types of gametes from the zygote. They are capital I, capital C, capital I, small c, small I, capital C, small I, small c. These are developed from one parental generation of F1. These are developed from the another parental generation of F1. These are crossed. These gametes are crossed to form F2 generation. In F2 generation, 16 types of zygotes are formed. The 16 types of zygotes expressed 16 types of phenotypic characters. From these 16 types of gametes, 13 types of ends, 13 types of ends containing white color hairs. White color hairs. See here it is. If capital I homozygotic condition or in heterozygotic condition, these all zygotes are containing capital I genes. So these are expressed white color hairs with leg RN. See, in last box of Punnett checkerboard, small I, small c, these are also expressed white color hairs with Plymouth horn. Finally, what we need from this Punnett checkerboard, we can observe phenotypic characters. What is the phenotypic character? Here it is in hens, white color hairs and colored hairs. White color hairs and colored hairs also identified. The white color hairs of hens are 13 and 3 R belongs to color. How to express these 3 R belongs to color? See here it is in 3 boxes. These are the 3 boxes. These 3 boxes containing 3 zygotes. The 3 zygotes are without dominant epistasis gene. Without dominant epistasis gene. Mean, see children, here it is small I, small I. The small I is recessive condition. The small I genes cannot act as a dominant genes. So they are in recessive condition. Which is dominant position? Here it is capital C, capital C. It is in homozygotic condition, or capital C, small c. It is in heterozygotic condition. So from these 3 zygotes, dominant epistasis gene is absent. So remaining dominant genes are expressed their own color or own character. What are the remaining genes? Here it is capital C. Capital C character is what? Colored. Colored hairs are developed by the capital C genes. How to express these colored genes? Without absence of dominant epistasis gene. Absence of dominant epistasis gene in these 3 zygotes. The dominant gene, normal dominant gene capital C is expressed their own color. What is own color? It is colored. Colored hairs are developed. So finally from this Punnett checkerboard, F2 generation phenotypic ratio is 9:3:3:1 change into 13:3. Finally, in dominant epistasis, the phenotypic ratio 12:3:1 or 13:3. 12:3:1 or 13:3. These are the dominant epistasis in hens with inhibitory genes. It is the second example